Ever wonder how scientists are actually rewriting the code of life?
Hmm.
It sounds like something out of science fiction,
Yeah,
but it's happening right now in labs all over the world
it is. Yeah.
with a little something called recombinant DNA technology. And lucky for us,
Yeah.
we've got a stack of research here, hot off the presses from biology notes online to help us break it all down.
It's pretty amazing stuff. Yeah.
So,
Imagine being able to take a gene from one organism and insert it into another.
whoa.
It's almost like you're copying and pasting text from one document to another,
Okay.
but with the very building blocks of life.
So we're not just talking about breeding plants or animals here.
Right.
This is about directly manipulating their DNA, right?
Exactly. It's like giving an organism a software update,
Wow.
providing it with entirely new capabilities.
Yeah.
And to really understand how groundbreaking this is,
Mhm.
we need to go back a few decades to the early 1970s.
Okay.
That's when scientists first started experimenting with combining DNA from different sources.
Now, I've heard about this. Wasn't there a scientist named Paul Berg who was one of the pioneers in this field?
Absolutely.
Yeah.
Paul Berg was the first to create a recombinant DNA molecule back in 1972.
Yeah.
But it was Stanley Cohen and Herbert Boyer just a year later who really cracked the code.
Wow.
They figured out how to insert these hybrid DNA molecules into bacteria,
Okay.
and even more importantly, how to get those bacteria to actually replicate with this new DNA.
It's remarkable, but I have to ask, how do you even begin to work with something as tiny and complex as DNA?
Right.
What are the tools of the trade for these genetic engineers?
Well, think of it this way. If you were editing a book,
Yeah.
you would need tools to cut out sections, paste in new ones, and ensure that all those edits were saved.
Right.
It's the same concept with DNA, just on a microscopic scale.
Okay, so what are the scissors and glue of the DNA world?
Scientists discovered these incredible enzymes called restriction enzymes.
Okay.
And these act like molecular scissors, but with an amazing level of precision.
Whoa.
They can recognize specific sequences of DNA and make incredibly clean cuts,
Okay.
which is essential for this kind of work.
So we've sliced up our DNA. We've got the gene we want to insert. What's next? Do you just get out the microscopic glue stick?
Not quite. We use another type of enzyme called DNA ligase, which acts as our genetic glue. It has the ability to form new bonds between those cut DNA fragments, essentially pasting them together.
So it's like a microscopic sewing machine just stitching those DNA strands together.
That's a great way to put it.
Wow.
And then, of course, you need a way to deliver this newly edited DNA into the target cell.
Okay.
And that's where vectors come in.
Vectors. This is where it gets a bit sci-fi for me.
Like CTs. Imagine a vector as a delivery truck carrying our precious genetic cargo to its destination.
Okay.
Often, these vectors are plasmids.
Plasmids. I've heard that term before, but to be honest, it's a bit fuzzy for me.
No problem. Think of plasmids like little self-replicating snippets of DNA found inside bacteria.
Okay.
They're like tiny USB drives that can carry extra genetic information.
Okay. That makes a lot more sense.
Good.
So we use these plasmids as our delivery trucks. We load them up with our sliced and diced DNA.
Precisely.
Yeah.
We insert our recombinant DNA into these plasmids, and then these modified plasmids are introduced into the host cell, which could be another bacterium, a yeast cell, even a plant or animal cell.
That's incredible. So we've successfully delivered our modified DNA into the host cell.
Right.
But how do we know if it actually worked?
Yeah.
How do scientists make sure that the host cell accepts the delivery and starts using those new genetic instructions?
That's a great question.
Yeah.
And it's actually a really crucial step in the process.
Okay.
Not every cell will take up the vector.
Right.
And even among those that do, not all of them will integrate the new DNA successfully.
Oh, okay.
It's kind of like sending out invitations to a party.
Okay.
You know, not everyone will RSVP.
Yeah.
And some might even lose the invitation altogether.
So how do you separate the party animals from the party poopers in the world of cells?
Well, scientists have these clever ways of screening and selecting for cells that have successfully incorporated the recombinant DNA.
Okay.
One common method is to use something called marker genes.
Marker genes. What are those?
Imagine attaching a bright fluorescent flag to your invitation.
Okay.
The ones who show up with the flag clearly got the message right.
Right.
Marker genes work in a similar way.
Okay.
They're inserted alongside our gene of interest,
Okay.
and they often code for easily detectable traits like antibiotic resistance, or a specific color change.
Wow, that's cool.
Yeah,
So you could grow the cells in a petri dish with antibiotics, and the ones that survive are the ones that took up the new DNA.
exactly.
Well, like a microscopic survival of the fittest.
It's a really powerful way to isolate those genetically modified cells.
Wow.
And once we have those, the possibilities are, I mean, they're really endless.
This is what I really want to dive into. The research mentions applications in medicine,
Oh, yeah,
agriculture.
absolutely.
It even hints at environmental uses.
Uh-huh.
Where should we start? What's got you most excited about the potential of this technology?
For me, it's the medical applications that are truly revolutionary.
Okay.
The ability to produce life-saving medicines like insulin using recombinant DNA technology has been a game changer for millions of people with diabetes.
That's right. And I read that before recombinant DNA technology, insulin had to be extracted from animals, which
Yeah.
was a much more difficult and less reliable process.
Exactly. And it wasn't just about efficiency.
Oh.
Animal-derived insulin, while effective, could sometimes trigger immune responses in patients.
Oh, wow.
Whereas human insulin produced using RDT is identical to what our bodies naturally produce.
Okay.
So it really reduces the risk of complications.
That's incredible. And this is just one example.
Right.
What other medical marvels are being developed with this technology?
Well, the list is constantly growing.
Wow.
We're talking about producing growth hormones for children with growth deficiencies, clotting factors for individuals with hemophilia, and even vaccines for deadly diseases like hepatitis B.
Okay.
The impact on human health has been profound.
It's like we've unlocked this whole new toolbox for treating diseases.
Yeah.
And it's not just about treating existing conditions.
Right.
The research also mentioned gene therapy, the potential to actually correct genetic defects at their source.
Precisely. Gene therapy holds immense promise for treating a wide range of genetic disorders, from cystic fibrosis to certain types of cancers.
Wow.
It's still a relatively new field, but the advances are coming rapidly.
Yeah.
We're really on the cusp of being able to rewrite the very errors in our DNA that cause these debilitating diseases.
That's mind-blowing.
It's incredible.
It's like science fiction becoming reality right before our eyes.
I know.
But what about the other applications you mentioned? The article also highlighted how RDT is being used in agriculture.
Absolutely. Recombinant DNA technology has played a pivotal role in developing genetically modified crops,
Okay.
or GMOs as they're commonly known.
GMOs? Now that's a term that often sparks debate.
It does, yes.
Can you tell us more about how RDT is used in this context?
Sure. And what some of the implications are.
Of course, one of the most well-known examples is the development of crops that are resistant to certain pests.
Okay.
Scientists have discovered genes in bacteria that produce proteins
Okay.
that are toxic to specific insects, but harmless to humans.
Okay.
By inserting these genes into crops like corn or cotton, farmers can significantly reduce their reliance on chemical pesticides.
So these crops essentially have their own built-in defense mechanisms,
Exactly.
reducing the need to spray harmful chemicals on our food and in the environment.
Exactly. And the benefits extend beyond pest control.
Okay.
RDT has been used to develop crops that are resistant to herbicides,
Okay.
making it easier for farmers to control weeds without harming the crops themselves.
Other genetically modified crops can withstand harsh environmental conditions like drought or high salinity,
Okay.
making them more resilient in the face of climate change.
That's incredible. It sounds like RDT is providing tools to tackle some of the biggest challenges facing agriculture today.
It really is.
But I know there are concerns surrounding GMOs as well.
Right.
What are some of the potential downsides that people are worried about? It seems like we're kind of, I don't know, standing on the edge of a whole new world when we talk about this tech.
Yeah.
But just like with any powerful tool, there are always trade-offs, right?
Right.
What are some of the concerns that people have raised about genetically modified organisms?
Well, one of the biggest worries is the potential impact on biodiversity.
Okay.
So, for example, if a genetically modified crop were to, say, cross-pollinate with a wild relative, those modified genes could spread in unpredictable ways,
Okay.
and that could lead to a loss of genetic diversity in those wild populations,
Okay.
potentially making them more vulnerable to diseases or environmental changes.
That makes sense. It's like introducing a new species that might outcompete the existing ones,
Right.
disrupting that natural balance.
Exactly.
Yeah.
Another concern is the potential for unintended consequences.
Yeah.
When you're making changes at the genetic level, it's so complex.
Right.
It can be incredibly difficult to predict all the potential downstream effects.
So while we might aim to solve one problem, we could inadvertently create another.
Exactly.
Right. Are there any examples of that actually happening?
There have been cases where genetically modified crops that were designed to resist certain pests actually led to the emergence of new, more resistant pests further down the line.
Wow.
It's this constant game of cat and mouse,
Yeah.
and it just highlights the need for ongoing research and really careful monitoring of these crops in the environment.
It sounds like a lot of responsibility rests on the shoulders of scientists and regulators to ensure this technology is used safely and ethically.
It absolutely does, and
Mm.
transparency is crucial.
Okay.
Consumers have a right to know how their food is produced,
Right.
and having these open and honest conversations about both the potential benefits and the risks of GMOs is essential.
Yeah, it's all about making informed decisions, both as scientists and as consumers, right?
Right,
Well,
exactly.
this technology has the potential to revolutionize our world from medicine to agriculture and beyond, but
It's amazing.
it also kind of forces us to confront some really complex ethical questions.
Absolutely.
Yeah.
That's the thing about a deep dive, isn't it?
Right.
You never know what you'll uncover.
Right.
But by grappling with these difficult questions head on, we can really harness the power of recombinant DNA technology for good, while also mitigating those potential risks.
It's a brave new world out there, and it seems like thanks to recombinant DNA technology, we're the ones writing the code.
It's pretty amazing.
This has been an incredible journey.
It has.
We've gone from the basic building blocks of life all the way to the future of medicine, agriculture, and really even the planet itself.
And this is just the beginning.
Right.
Who knows what incredible breakthroughs are waiting for us in the years to come.
I can't wait to find out. Until next time, keep exploring, keep questioning, and keep diving deep into this fascinating world around us.